So, yeah, good afternoon, everybody. Mm. Okay, so let's start today's lecture. So I know now that you have covered the skeletal system in your labs. So now you're prepared for today's class. And from now on, we are going to be much closer to the lab, the subject, the lab material. So, hola. So we're close to Halloween. So to study the skeletal system is very appropriate. And it's a very interesting system. You see here, it provides movement, but it has many other different functions. So what are the functions of the skeletal system? Some are very obvious. So it supports soft tissue. Just think about the cranium, how many bones it has and how strong it is, and its main function is to protect the brain. Also, for example, the rib cages also protect the lungs, for example. So it protects internal organs from injury and from in the brain, it provides a very, very strong cage to prevent the soft tissue of the brain from being injured. It also provides body movement. And why is that? Because we have the muscles and they are attached to the bones, and that's the bones provide leverage. So the muscles are attached to the bones via these connective tissue structures that are known as tendons. So tendons are the means by which the muscles are attached to bones. And two bones are connected to each other by ligaments. So tendons and ligaments are connective tissue. Tendons are used to connect muscles to the bones, and ligaments connect two bones. Now, this is a very interesting function of the skeletal system. And it provides homeostasis, mineral homeostasis, and mostly calcium. So the calcium levels in the blood are regulated through mechanisms that either deposit or remove calcium from the bones. So the bones are, from all the structures in the body, the structure that contains and most calcium. And when the blood levels go down, then there are cells, we will discuss this, that are part of the bone and that release calcium from the bone. If the blood levels are too high, then there are another type of bone cells known as osteoblasts, and they deposit calcium on the bone. So you have these two different types of bone cells, osteoblasts and osteoclasts. The osteoblasts are the architects of the bones. So they deposit calcium on the bones. The osteoclasts, which are gigantic cells, they remove calcium from the bones when the blood calcium levels are too low. So you have these two different types of cells that work together to maintain the calcium levels, the homeostatic, the calcium levels in the blood. So another very important function of the bones is they store the stem cells for all cells that are in the blood. So white blood cells, red blood cells, and platelets. The stem cells, the precursors, are stored in the bone marrow that is stored in spongy bone. So it is within these storing locations in the spongy bone where hemopoiesis occurs. What is hemopoiesis? Hemopoiesis is the maturation of the different types of blood cells, white blood cells, red blood cells, and platelets, that occurs or starts in the bone marrow, in the red bone marrow. So you see that we have around 1 billion white blood cells, 200 billion red blood cells, and 100 billion platelets. Now, from all of these, the red blood cells are the ones that are most stable. They're around for 120 days. The white blood cells, they are substituted around every 20 days, and so are the platelets. So we are continuously relying on these precursors of these different types of blood cells that are stored within the trabecula of the spongy bone. Also, the bone stores the yellow bone marrow. And what is the yellow bone marrow? The yellow bone marrow contains fat. And this fat is a very important tissue, energy storage. You know that it's made up of fatty acids, and the fatty acids contain many, many carbons that are linked by covalent bonds. And when you break apart, when you have the enzymes that break apart these covalent bonds, you have energy being produced. And this occurs mostly in the mitochondria. So you see that there's a lot of functions, very important functions, that are related to the skeletal system. The skeletal system is not only important for protection and for providing movement, it's important for maintaining calcium and phosphate homeostasis, and particularly calcium is very important. It's also the site where the red bone marrow is stored. So this is where hemopoiesis starts, where the generation of different blood cells occurs from the precursors. And then it also is a site where there's storage of energy like fatty acids in the yellow bone marrow. The yellow bone marrow is stored in the long bones. Okay, so these are the major functions of the skeletal system. Okay, now the bones in our body can be classified depending on their shape and depending on their structure. So there's many different shapes of bone. And it's amazing how the body and the DNA can regulate eventually where these bones with the different shapes are going to end up in our body. So the sternum has this flat bone appearance. Then the vertebrae have a very sophisticated and intricate shape, and they are the ones that protect the spinal cord that comes through the vertebra. Then the patella, which is this bone that is located in the knee, and this has a special shape that's a sesamoid shape. Then we have short bones and long bones. And the long bones, they have a cavity in the middle. They're not all compact. That cavity is known as the medullary cavity, and that's where the yellow bone marrow is stored. Now, how about the structure? So this is shape. How about the structure? So we classify the bones in our body, depending on structure, into two major groups. We have compact bone and spongy bone. And you can see that most of the bones are compact. 75% of the bone structure in our body is compact and 25% is spongy. Now, it's a very, very interesting structure. If you look at the spongy bone, so it's like a sponge. What is a sponge? A sponge is a structure that has openings, it has holes. And here, it has also holes, the spongy bone. It has these structures that are bone, but these structures that are known as trabecula, they surround these cavities. And it's within these cavities that the red bone marrow is stored. So the red bone marrow, remember, it has the precursors of the blood cells. So these precursors are stored in these cavities located in the spongy bone. The compact bone, you see, as its name indicates, it's compact. It doesn't have these cavities, but it surrounds the medullary cavity in the long bones. And the medullary cavity is where the yellow bone marrow is stored. So this is a cross section of a bone, and you can see the yellow bone marrow in the medullary cavity. Then you see the spongy bone, and these structures here represent the cavities where the red bone marrow is stored. And then you have the trabecula. That's where these are made of bone tissue. And you have the compact bone on the outside. Now, this compact bone is protected by a layer of connective tissue that we call periosteum. Okay, so let's continue. Okay, so this is another other images representing the spongy and compact bone. So this is the femur, the head of the femur. So you see right here, this is spongy bone. So you see the trabecula, you see that it's not compact, and this forms cavities where the red bone marrow is stored. But you see that the outside is compact bone, and it surrounds the medullary cavity where the yellow bone marrow is stored. So here you see the spongy bone, you see this pink, this is a histological preparation of a spongy bone, and the pink structures are the trabecula. So this is real bone tissue that surrounds these cavities that contain the red bone marrow. And here you see the compact bone. What is interesting about the compact bone is that the cells, the bone cells, are trapped in this very compact matrix, and they cannot move. They're stuck there. And these are known as osteocytes. So they're the most mature cells that go from osteoblasts. They then transition to osteocytes. Okay, so here's another image, another slide showing the medullary cavity with a compact bone on each side. What type of cell is in the middle of an osteon? So is it osteocytes? Yes, correct. So here's the compact bone surrounding the medullary cavity where the yellow bone marrow is stored. So in adults, these they have mostly yellow bone marrow. And in children up to 5 years old, they have mostly red bone marrow. So you see here the trabecula that makes up the spongy bone and these cavities where the red bone marrows are stored. And this is another image showing the yellow bone marrow and the red bone marrow that contains all the precursors. So we call it red, but don't confuse it that it only has the precursors of red blood cells. No, it has the precursors of white blood cells, red blood cells, and platelets. Okay, so let's look at the long bone and the different structures that make up this long bone. So at each end, we call this area of the long bone the epiphysis. So there's two epiphyses, one at one end and the other at another end. And now immediately you see that there is some cartilage here that is protecting the outside of the epiphysis. This is on each side, and this is called the articular cartilage. So when someone says that they have arthritis and they have a lot of pain, that's because one of the reasons is that the articular cartilage is worn out. So now you have two bones rubbing against each other. There is no articular cartilage, or the articular cartilage is damaged. So there's no protection. And that's what causes the pain. So the articular cartilage is a cartilage that protects the epiphysis, on each side of the long bone. Now, in between the two epiphyses, we have the diaphysis. So the diaphysis contains compact bone and contains the medullary cavity that is here represented by this orange ellipse. Okay, so on the outside of the diaphysis, you have connective tissue that's known as periosteum. And why is the periosteum important? Because this connective tissue that protects the diaphysis is where the tendons and ligaments attach to. So the muscles and one bone is attached to another through ligaments or tendons for the muscles, and they connect to this periosteum. So now there's a last very important area of the long bone. So we talked about the epiphysis and the diaphysis. Now there's an area here between the epiphysis and the diaphysis that is known as the metaphysis. So this is this area here that is not really shown very clearly. But why is the metaphysis important? The metaphysis is important because this is the area that allows the bone to grow in length. So it has what we call the epiphyseal plate, and this is the last area of the whole bone to become calcified. So there's a lot of activity that goes on in this metaphysis in these areas, and the effect is to allow the bone to grow in length. Now, another lining that I didn't mention is the endosteum. So the endosteum surrounds the medullary cavity. And the endosteum, it's also connective tissue, and it has osteogenic cells. And I will explain what those are. So you see that there are many different areas and structures in a long bone. Remember that the metaphysis is the area between epiphysis and diaphysis, and this is where there's a lot of activity, a lot of mitosis and so on, that allows the bone to grow in length. So here is a magnification, let's say, a cartoon, that shows the articular cartilage here in purple. And this is very important to protect the epiphysis of these two bones and prevent the bone from rubbing against each other. So, but there's a lot of wear and tear that this cartilage, if it's not maintained in a perfect condition, in a healthy condition, then it wears out, and this allows the bones to rub against each other, and this leads to a lot of pain. Now, this is a very complex type of joint that we will discuss later, but you see here that there's this fluid that is a synovial fluid, and we will we'll talk about later. Here you have a muscle and you have a tendon, and the tendon is going to attach to the periosteum. But here what I wanted to point out is the articular cartilage that protects the epiphyses of the bones, preventing them to rub against each other. So it provides a cushion. Okay, so here we have the periosteum and the endosteum. So this is connective tissue. And you see that the periosteum is present in the outside of the diaphysis of the compact bone. And this is where the ligaments and tendons attach to. It also has blood vessels, and it contains the osteogenic cells. So genic means that they lead to something. These cells are bone cells that have the capability to undergo mitosis, and they give rise to these cells that are the osteoblasts. And remember, at the beginning of the lecture, I mentioned that the osteoblasts are the architects of the bone. They're the ones that deposit calcium on the bone. Those are the osteoblasts. And the precursors of the osteoblasts are osteogenic cells, some of them located in the periosteum, others located in the endosteum. So the endosteum is the layer of connective tissue that protects the medullary cavity. So you see here, this is an osteogenic cell, so it's a precursor of osteoblasts. Okay, so now let's talk about the different types of cells that make up the bone tissue. So we'll start with the osteoblasts. So these are the architects of the bone. These are the ones that deposit calcium on the bone and make the bone more dense. Okay, so what is their origin? What are their precursors? The precursors are the osteogenic cells. These cells undergo mitosis, and they can give rise to osteoblasts. The osteoblasts lose the capacity of undergoing mitosis. So they are derived from osteogenic cells, and the osteogenic cells are derived from mesenchyma. So you have this transition going from mesenchyma to osteogenic cells or osteogenic cells, and these are then going to differentiate into osteoblasts. Now, the osteoblasts are very important. They are the architects. They provide the means to deposit calcium on the bone, and they form material known as osteoid. So this osteoid is secreted by the osteoblasts. When this osteoid becomes calcified and then traps the cells, the osteoblasts now transition to the final mature step, which is the osteocytes. So 90 to 95% of the cells in the bone are the osteocytes, and they are trapped in this matrix that has been calcified. And the matrix is derived from the osteoid. So the osteoid is still not calcified, it's not hard yet, and it's produced by the osteoblasts. So once this osteoid becomes calcified, then the osteoblasts transition to the last step, which is the osteocytes. So here you see a histological preparation of the bone tissue, and you see the osteoblasts here, and then they will produce the osteon. Once the osteon becomes calcified, then the osteoblasts transition to osteocytes and become trapped in this calcified matrix. Okay, so here in this upper layer, upper level, you see the transition from osteogenic cells to osteoblasts and to osteocytes. So the osteogenic cells, their function is to divide and provide more osteoblasts. So it's thought that the osteogenic cells, for example, have receptors for estrogen, and they are activated by exercise. So that's why exercise can make the bone more dense, because the osteogenic cells become more active, they make more osteoblasts, and the osteoblasts deposit more calcium, and therefore the bones become more healthy and the more dense. Now, the osteocytes, their main function is to maintain the bone tissue. Now, let's look at the other type of cells in the bone. So these up here, I'm a little confused. Where do osteogenic cells come from? The osteogenic cells come from mesenchyma. So mesenchyma is the precursor of the osteogenic cells. Okay? So you have mesenchyma that differentiates into osteogenic cells, and then you have this progression. Okay. So now, so these are the architects of the bone. Now, if there is low levels of calcium in the blood, where do you get calcium from? You get calcium from the bone. And how? So here come the osteoclasts. So you see that the osteoclasts are these gigantic cells. They have many nuclei, and they have these projections, and also they are very rich in lysosomes. So the origin of the osteoclasts are not the osteogenic cells. They come from monocytes. They come from precursors of white blood cells. So these monocytes, which are precursors of white blood cells, differentiate into osteoclasts. Some of these fuse to each other, and that's why they make these gigantic cells that are the osteoclasts. And you see that they have these projections. And what is the function? The function is that they release enzymes that will release calcium from the bone. So you see here compact bone with the osteocytes, which are much, much smaller than the osteoclast. So the osteoclast, when it becomes active, it releases enzymes that now chew up the bone matrix to release calcium. So you have these two very different cells or lineage of cells. These osteoblasts, they make the bone tissue by producing osteon that eventually will become calcified. And these, the origin is in the mesenchyma. And then you have the osteoclasts. They'll start as monocytes. So we could consider them a type of white blood cells that has differentiated into bone cells. So these are the osteoclasts. And this area that has this projection is known as the ruffled border. So here you have a release of enzymes that chew up the bone matrix to release calcium. So I think that this is very interesting. You don't think about bone maintaining calcium levels, the homeostasis of calcium in the body. And this is because of the action, the opposite action of these two different types of cells, the osteoblasts and the osteoclasts. Okay, so here is again a histological preparation. Where you have here is a osteoblast, and just compare the size of an osteoblast with an osteoclast. So this is a huge cell that has many nuclei and has this ruffled border with projections that release those enzymes that will break down the calcium to release the calcium from the bone. Okay, so again, you have here the osteoclasts. The origin of the osteoclasts are the monocytes, which are a type of white blood cells. And the process by which the osteoclast releases calcium from the bone is known as resorption. It's not absorption, it's resorption. So remember this word, resorption. So what is resorption? Is the process by which the osteoclasts release calcium from bone matrix. So here again, you see the osteoclast. This is the marrow cavity, the bone marrow cavity. And now you see that the osteoclasts form what we call this resorption bay. So they start chewing and chewing, and they make these areas that are sort of empty of cells, that they are filled with calcium. Okay. So we talked about the cells in the bone, the osteoblasts and osteoclasts. But what makes up the bone matrix? So the bone matrix, where the osteoblasts and osteoclasts are located, is made up mostly of calcium. So it's made up of calcium salts. So the most abundant calcium salt that is a component of bone matrix is known as hydroxyapatite. So hydroxyapatite has this structure. It's a molecule that's highly enriched in calcium ions and also phosphate. And this is hydroxy. So you see that it's highly, highly enriched in calcium. So the bone matrix is made up of salts that are enriched in calcium, such as hydroxyapatite, but also collagen fibers. So why is this so important for the bone matrix? So the salts, their main function is to establish some type of hardness in the bone. So the bone is not soft tissue like fat or skin. The bone is hard, and that hardness is provided by the calcium or mineral salts. But at the same time, the bone has some tensile strength. What does that mean? That means that every time you fall, you don't break a bone, because the bone can withstand some kind of stress. If you fall with a big fall, then you can break a bone, you can fracture a bone. But for example, if I hit my arm against the table, I don't break the bone. It's not like china. If you throw a plate made up of china on the floor, it will break into 1,000 pieces. But the bone, the matrix, because it has these collagen fibers shown here, it has this tensile strength, which means that it's capable of withstand some stress. So you have this combination of mineral salts and collagen fibers that are very important for the properties of the bone matrix. Something. So, okay, so we can talk about bone metabolism because we have these two cells. One makes bone, so it's the osteogenesis. Genesis is to produce, to make. Osteo, every time you see this prefix, that means it's related to bone. So you have osteoblasts that make bone, that they are playing a function in osteogenesis. So the osteoblasts produce the osteoid, which is not yet calcified, and then once it becomes calcified, it gives rise to bone. But you also have osteolysis. So the break part, the breakdown of the matrix, and that's carried out by osteoclasts by a process known as resorption. So here in this cartoon, you have the osteoblasts that line up very nicely, and they produce the osteoid. And over here, you have the osteoclast that is resorption the bone tissue. So you have these two types of cells, one that plays a role in making the bone tissue in osteogenesis, and the other one that plays a role in breaking down the bone matrix, and that is carried out by the osteoclasts. No questions today. Okay, good. Okay, so now I want to transition to how is bone formed. So the bone formation is known as ossification. So the question is, does resorption only happen if there's not enough calcium in the diet? Yes, but it's also regulated by hormones and also occurs to increase the thickness of the bone. So you will see it's not only regulated by calcium levels in the blood, it's also regulated by the bone growth, for example, and by hormones. Okay, so around a fetus around 6 week old, the skull and the skeleton is still made up of cartilage. So the ossification starts around the 6th or 7th week of embryonic life. See, this is a 10-week-old human fetus, and it has already, ossification has already started. Ways in which this the ossification can occur. So there are two types of ossification: the intramembranous ossification and the endochondral ossification. So they're very different, and they lead to bones with very different shapes. So the intramembranous ossification, it starts by forming a core. So let's say you're playing with Play-Doh. So you have a ball of Play-Doh, and now you want to mold it into a bone. So you can stretch and so on and give rise to a bone. That's what the intramembranous ossification starts. It starts with the core that then grows in different directions, leading to different shapes, bone shapes. And this occurs, the bones that differentiate through or ossify through intramembranous ossification of, for example, the skull, the mandible, and the clavicle. So these are examples of bones that form by intramembranous ossification. So you start with the core, and then it stretches in different directions, grows in different directions to give rise to different shapes. How about the endochondral ossification? Now, this is very different. It starts with a frame or a mold that's made up of cartilage. So it doesn't start as a core. It starts as a frame, as a mold that is made up of hyaline cartilage. And then it starts differentiating inward into bone tissue. It starts ossifying. So this endochondral ossification starts with a predetermined shape that's made up of hyaline cartilage. So example of bones that form, that ossify via endochondral ossification are the humerus and the femur. Okay, let's look into more detail into the intramembranous ossification. So the one that starts with the core. So the first thing you see here, the mesenchymal cells, which are those cells that are the progenitors to the osteoprogenitor cells. So this is known as an ossification center. that starts within a layer of mesenchyma. So what happens? You have the osteoprogenitor cells, here shown in blue, and they start dividing by mitosis, and then eventually they differentiate into osteoblasts. And here is the osteoid, shown in light orange. So this is an ossification center that starts occurring in a layer of mesenchyma. Then this osteoid matrix now becomes calcified, and you have the osteocytes within the calcified bone, and you still have the osteoblasts surrounding this calcified bone matrix. So this is a transition. Then you have blood vessels invading this area, and they give rise also to the spongy bone, to the spongy bone that contains the bone marrow. But you see on the outside, you have the osteoblasts that will give rise to compact bone. So here are the transition where you start with an ossification center that will eventually transition to calcified bone. And then you have spongy bone and compact bone forming from this ossification center, and again giving rise to a bone with a specific shape, with the periosteum on the outside, the compact bone, and the spongy bone in the middle. So here you have another histological preparation. So this very dark area is the calcified bone. So here the bone matrix already has hydroxyapatite and collagen fibers. So it's already formed, the matrix is already formed. Here you have the osteoblasts, and here in this translucent pink area is the osteoid. Once it's calcified, it forms this calcified bone. So in this intramembranous ossification, there's no model, there's no frame. It starts from a core. So sometimes this intramembranous ossification can start in areas that it shouldn't occur. And this can lead to heterotopic bone formation using this intramembranous ossification. Remember that it starts with the primary ossification center. So this ossification center can start in areas that it should not be there and lead to this deformed kind of ossification that's known as heterotopic. Heterotopic because it's outside the skeleton, and it's heterotopic bone formation. This can be very painful because sometimes it can be formed within muscle. So you see here that there's bone formed in muscle tissue, and this is known as myositis ossificans. When you see the prefix myo means muscle. So ossificans, that means that there's bone formed within the muscle. So you have bone tissue, this hard bone tissue formed within the muscle. And you can imagine how painful that can be. Now let's switch to the endochondral ossification. Now look up here the timeline that endochondral ossification goes through. So it starts at week 9 or 10, and then it continues month 3 at birth. So this is embryonic or fetus life. So at birth, and then it continues to adolescence. So it's a very long period through which bones are formed through endochondral ossification. So let's go through the different stages. So remember, you have mesenchyma. In the mesenchyma, you have chondroblasts. So what are chondroblasts? Chondroblasts are cells that can form cartilage. So you start with a frame that is made up of hyaline cartilage. This is represented here in blue. Then you have the ossification centers. Just like in the intramembranous ossification, you start with an ossification center in the middle of this mold, of this frame. So you have the osteoblasts being formed. So this is the primary ossification center that forms in the middle of this hyaline frame. So you can imagine, or you can foresee, that these extremities are going to give rise to the epiphysis, and this area here is going to give rise to the diaphysis. So this first primary ossification center starts forming in the middle of this frame, and then you have some of the bone will be deteriorating, and it starts forming the medullary cavity. So this is the primary marrow cavity. So remember that within the diaphysis, you have this medullary cavity that stores the yellow bone marrow. So from the primary ossification center, some of the bone, some of the cartilage starts deteriorating, and it starts forming the marrow cavity. Now the next, so primary marrow cavity, the next step is the formation of spongy bone, and then you have new ossification centers forming at the site that is going to give rise to the epiphysis. So here now you have spongy bone being formed in the periosteum, and now this center area that gives rise to the diaphysis continues to form and leads to the medullary cavity. And then on each side, you have the secondary ossification centers where osteons and osteoblasts start working to make bone. Remember, all this starts with hyaline cartilage, right? The blue represents the hyaline cartilage. So you still have hyaline cartilage at the epiphysis, while the diaphysis now is becoming ossified. So you have two secondary ossification centers, one at each end. And then you have the ossification continues at these two secondary ossification centers. And now you see here, between the diaphysis and the epiphysis on each side, you see still some cartilage. And this is what makes the epiphyseal plate cartilage. Yeah, the pericardium starts forming parallel with this, this whole process. Here, remember, it's the articular cartilage that remains there, and the periosteum starts forming as you go along. You see here, as you go along in this differentiation. So, are epiphyseal lines and plates the same? No. So the epiphyseal plate is when you still have the ability of the bone to grow. Once the epiphyseal plate becomes epiphyseal line, then the metaphysis is gone. There is no more bone growth. So the epiphyseal plate, there's still growth possible. Once you reach the epiphyseal line, that means that this area has no more cartilage. Everything is bone, and there's no way of the bone to grow more. So once you go from the plate to a line, when you get to the line, the bone stops growing. So epiphyseal plate, you can still have growth because you have still some cartilage. Once the epiphyseal line is formed, then there's no more cartilage and the bone reaches its size. So you see here the articular cartilage and the epiphyseal plate. So the epiphyseal plate still has cartilage. It still shows this blue area. Of course, it's not blue in real life. Okay, so let's look now more in detail in a histological preparation. What happens here at the epiphysis? So this is the, let's say, the epiphyseal plate. So it's also known as the metaphysis. And you have here cells that still can undergo mitosis. And because they can undergo mitosis, the bone can grow this way and that way. So this is the secondary ossification center. You would have another one at this end. So while the epiphyseal plate is formed, while you have cells that can undergo mitosis, this bone can still grow. And sometimes it's not very good idea to operate on bones in children because they still have epiphyseal growth, they still have the epiphyseal plate. And if you damage this epiphyseal plate, you prevent growth. So is the epiphyseal plate found only on the proximal epiphysis? Yes. No, it's found in both epiphyses, the proximal and the distal. Here I'm just showing half of it, but this happens here and here. I'm just showing this area. So the epiphysis, the metaphysis, and diaphysis. So here you see the metaphysis yet with epiphyseal plate. So there's still cartilage here. There's still cells that can undergo mitosis. When we develop the epiphyseal line, do we still have osteoblasts and clasts, or is just osteocytes? So the notion that once the bone is formed, there are no, we don't need osteoblasts and osteoclasts. We do, because the bones are always changing in shape. They're very dynamic, and we always have osteoblasts and osteoclast activity. Also, when you have a fracture, you will need osteoblasts and osteoclasts to, so that the bone grows back to its shape. Okay, so this is a good site that explains the intramembranous and endochondral ossification. Okay, so we went through how the bone grows in length, but how does the bone grow in thickness? So the bone growth, you can have it in length, that's interstitial growth, but it can also grow in thickness. So the bone of a child is not so thick as a bone of an adult. So it grows in thickness, not only in length, but also in thickness. So the growth in length is known as interstitial growth, and the growth in thickness is appositional growth. So let's look at interstitial growth. So this has nothing to do with ossification. Now we're talking about how do bones grow. So it's a separate process. So interstitial growth. So this is dependent on the epiphyseal plates. So you see here the epiphyseal plates. This is an X-ray. So of a hand that still can undergo growth because it shows the epiphyseal plate where you have cells that can undergo mitosis. So it's thicker. Once these epiphyseal plates lose that layer of cells that can grow and undergo mitosis, then you see only the epiphyseal lines, and there's no growth that continues. So let's look at the metaphysis. This, at the metaphysis, is where the bone can grow. Interstitial growth occurs at the metaphysis. So the metaphysis is the area in both epiphyses where the bone growth occurs. So what type of cells are at the metaphysis that allow the bone to grow? So it's all based on cartilage. So the cells that are involved in this bone growth are known as chondrocytes. So here you have a layer of chondrocytes that they are just resting, they're not active. But then the layer after that, you see mitotic figures. This would be an anaphase, and so on. There's no metaphase or anything, but you can see mostly anaphase figures here. So this is where the cartilage, the chondrocytes are proliferating. So they are dividing. So they create more and more proliferating layers. Now, the next layer are the hypertrophic cartilage. So these chondrocytes grow in size, not in number, now in size. That's the hypertrophic. So they're growing in size. And then eventually they die out and they are substituted by osteoblasts. So that means that this cartilage now becomes ossified because now you have the bone deposits and you have the osteoblasts. So it's here. Remember that the osteoblasts cannot undergo mitosis. So they don't really contribute to bone growth. They contribute to bone ossification, but not to bone growth. So the bone can grow as long as these chondrocytes, as these proliferating layers of cartilage, can undergo mitosis. So this process is responsible for the interstitial growth. So you see here the metaphysis, and it continues to grow. The cartilage divides, proliferate, and the growth can go in this direction and in this direction. What about appositional growth? The growth in thickness. So here you see a cross section of one of these long bones, the femur. So in an infant, you see that the medullary cavity, the diameter is, let's say, quite small, and it has a layer of compact bone around it. Now you need the joint collaboration between osteoblasts and osteoclasts. So the osteoblasts keep depositing bone, as you see here. The thickness of the compact bone is increasing, but at the same time, you have the osteoclasts that are chewing the inside of the compact bone to increase the diameter of the medullary cavity. So you have the combined action of osteoblasts that make the compact bone layer thicker and the osteoclasts that increase the diameter of the medullary cavity. So you have bone deposition by the osteoblasts and bone resorption by the osteoclasts. So here, while for the interstitial growth depends on this proliferating cartilage, here the appositional growth depends on the combined effort of osteoblasts and osteoclasts. Okay, so we go to bone remodeling, and this will answer the question where do we still have osteoblasts and osteocytes and osteoclasts? Anyway, this is known as bone remodeling. That happens in adult life all the time. This is a very, very active kind of process where you have the osteoblasts depositing bone tissue, bone matrix. So this is represented in this area here, area 2. And then you have the osteoclasts that are chewing up to lead to the proper shape of the bone. So the osteoclasts destroy the bone, the old bone, and the new bone is generated by the osteoblasts. Okay, so what does exercise do? So exercise strengthens the bones. It increases the density of the bones. There's more calcium deposited on the bone. So this is regulated by the Wolff's law, which says that bone is restructured in response to stress. So if you lift up weights or if you play tennis or any kind of exercise, that exercise will activate the osteoblasts and cause them to deposit more calcium, and therefore your bones become more dense. So Here's a question. Would a good assumption be that the length and width of bones will be determined by the DNA of the individual? No, not really, because there's many factors, environmental factors, that can also contribute to the size of bones. So it could be that there's some genes that will determine, like in certain areas or certain families, individuals are shorter than in other families. But this regulation of size is not only dependent on genes, it's dependent on diet and all kinds of environmental factors. So here you see lifting weights is a very good way of preventing osteoporosis, for example, because in osteoporosis, you have less calcium in the bones. So if you do these weightlifting exercises, you will activate the osteoblasts and prevent the bone matrix from being destroyed. So what happens to individuals that are not exercising? That means that the osteoclasts take over and the bones become less dense, and also in astronauts, because there's no gravity. So the osteoblasts are not being active. So astronauts, they lose 1% of bone matrix per week. So what causes people to get shorter when they get older? So you know that the vertebra, there's intravertebral discs and between vertebrae. And it's thought the reason why individuals are shorter as they age is that the structure of these intravertebral discs loses its properties. That could be one explanation. Okay. Why do bones crack? Is it good or bad? Cracked, you mean in terms of fractures, or when you make noises? That's because of the ligaments, and you shouldn't do this because it makes the ligaments less flexible. I don't know if that's what you're referring to. So, as I said, not only stress regulates bone growth, but there are hormones that regulate bone growth. So before puberty, what are the hormones? Yes, that's that noise that you hear when you crack your bones. It's due to ligaments. So prior to puberty, there's some hormones that regulate bone growth. Human growth hormone. It's released by the pituitary gland. Actually, it's given to individuals if they don't grow enough. This could be a way of increasing the growth. This is clinically approved. So insulin growth factor produced by the bone and liver, and thyroid hormone and parathyroid hormone, which we are going to focus on. These mostly control calcium levels, and insulin in the pancreas. So prior to puberty, all of these factors control growth. At puberty, there's a sudden production of sex hormones. Ovaries produce estrogen and testes produce testosterone. So estrogen seems to have an effect on the epiphyseal plate. So they make the epiphyseal plate close faster. So what does that mean? That means the epiphyseal plate will become ossified faster because of estrogen. And that's why biological females sometimes are shorter than biological males, because the testosterone does not have this effect on the epiphyseal plate. Okay, now let's look at how calcium level, calcium homeostasis, how is it regulated in our body. So we're talking about calcium homeostasis. So the normal calcium in the blood is between 8.7 and 10.4 milligrams per deciliter. Now, these numbers are not exact. If you read different books, they will have a different range, but this is more or less in this range. So there are two conditions, hypercalcemia and hypocalcemia. Hypercalcemia, that means that there's too much calcium in the blood, and this can lead to depression of the nervous system, so the nervous system won't work as well, and it can lead to cardiac arrest because the nervous system is not working properly. So hypocalcemia is when there's calcium deficiency. And in this case, the nervous system will be more excitable. And I will explain this. So when there's low calcium levels, the nervous system is more excitable. When there's less, when there's more calcium, it's depressed. And when there's low levels of calcium, because the nervous system is so excitable, it can lead to tetany. So here's the range of phosphorus in the blood, but this is not so important. It is important, but I want to focus on calcium. Tetany is when your your body is kind of, the muscles are continuously active. So that is a condition known as tetanus. There's no relaxation. So vitamin D plays a very important in regulating calcium levels. So when we are regulating calcium levels, you have to think about these three different structures: the bone, the kidney, and the gut. So what I'm going to tell you, the different hormones and conditions, they all regulate the calcium levels at the bone, at the kidney, and at the gut. So vitamin D, you know that it's produced at the skin, the first step. Then there's a step between the skin and the kidney. There's the vitamin D3 goes to the liver. It's converted into calcidiol, and then the calcidiol goes to the kidney, and it forms the active form of vitamin D, which is known as calcitriol. So what does vitamin D do? How can it affect calcium? So vitamin D allows calcium to be deposited on the bone. So it prevents, it slows down the osteoclasts. It reduces how much calcium is going to be excreted by the kidney. So here you slow down osteoclasts, so there's more calcium remaining on the bone. You slow down the excretion of calcium by the kidneys, and you increase absorption of calcium by the gut. So that's the function of vitamin D. Now let's go to thyroid gland. So the thyroid gland is here in the neck region, and it has this butterfly kind of shape. And behind the thyroid, we have 4 parathyroid glands. So each of these produces hormones that are important for regulating calcium homeostasis. So let's start with the thyroid gland. So within the thyroid, we have 2 types of cells in a simple way. The follicular cells that form these circles, and these follicular cells produce a hormone known as thyroxine. So what does this hormone do? It increases the activity of osteoblasts. Then these other cells that are outside the follicles are known as parafollicular cells, and they release calcitonin. What does calcitonin do? It does more than thyroxine. It also increases osteoblast function. It decreases osteoclast function, and it increases the excretion of calcium by the kidney. So when is a thyroid activated? These cells in the thyroid are activated when there's too much calcium in the blood. So when there's hypercalcemia, these cells in the thyroid gland are activated. The thyroxine increases activity of osteoblasts, so there's more calcium removed from the blood and deposited on the bone. So remember, what you want to do is decrease calcium levels. Then the calcitonin, what it does, it also activates osteoblasts so that there's more calcium being deposited on the bones. It decreases the activity of osteoclasts, so there's less calcium being removed from the bone, and it increases calcium excretion by the kidney. So remember, the thyroid is activated when there's too much calcium in the blood. So the effect of these two hormones is to lower blood levels of calcium to the homeostatic range, to this range. What about the parathyroid? So the parathyroid is very, very important to maintain calcium levels. It's more important than the thyroid. So the parathyroid glands, there are 4 of them, so 2 pairs, and they are behind the thyroid. And what they produce is a hormone known as PTH, or parathyroid hormone. So the parathyroid glands are activated when calcium levels are low. So the thyroid gland is activated when calcium levels are high, under hypercalcemia. The parathyroid hormone is activated when calcium levels are low, so under hypocalcemia. So what does the parathyroid hormone do? So remember, now you want to increase the levels of calcium in the blood. So you prevent the kidney from excreting calcium so that there's more calcium staying in the blood. So you slow down the excretion of calcium by the kidneys. You increase the production of calcitriol by the kidneys. And you increase osteoclast activity. Remember, thyroid hormone lowers osteoclast because you don't want more calcium to go into the blood. The parathyroid hormone, that's the opposite effect. You increase osteoclast levels because you want more calcium to go into the blood. Okay, so in the last few minutes, I want to review all this that I said, which is a little bit confusing when you first encounter all these mechanisms. So let's say now that you have high levels of calcium. So we're talking about hypercalcemia, so that the concentration of calcium in the blood is above 10.4 milligrams per deciliter. So the thyroid will release calcitonin. What does calcitonin do? It increases the excretion of calcium by the kidneys. Because it's high, you want to get rid of calcium. So one way is to have the kidneys to excrete calcium. So vitamin is called so close to vitamin C in its active form because its function. Well, vitamin D and vitamin C are different. Now we're talking about vitamin D. It plays a very important role in calcium homeostasis. So you want more calcium to be excreted by the kidneys. So what about the osteoclasts? So you have too much calcium in the blood. So you want to slow down the osteoclasts so that there's no calcium being removed from the bone into the blood. And you want to activate osteoblasts, so more calcium is deposited on the bone. And now you to decrease parathyroid hormone activity and vitamin D effect. Okay, so the major players here to lower calcium levels is this hormone calcitonin produced by the thyroid. What about if the calcium levels are too low, and now you need to increase blood calcium levels. So now this is where the parathyroid hormone is very important. So remember, the parathyroid glands are these 4 small glands behind the thyroid, and they play a very, very important role in calcium homeostasis. So if the blood calcium levels are low under hypocalcemia, there are cells, receptors on the parathyroid that become activated, and they increase the release of parathyroid hormone, PTH. What does it do? It acts at the kidneys. So now you want to prevent calcium excretion. You want more calcium to remain in the blood and not be excreted by the kidney. So now you want to activate osteoclasts. Why? Because you want more calcium to be removed from the bone and to go into the blood. So here you have parathyroid hormone acting at the kidney, acting at the bone, and also at the gut. What does PTH and calcitriol or vitamin D do at the gut? It increases the rate of absorption. So that means that there's more calcium going from the lumen of the gut into the blood. So this is a very, very important mechanism that is responsible for the homeostasis of calcium in the blood. And the most important hormone is the PTH or parathyroid hormone in conjunction with vitamin D. Okay, so I think I've stopped just one more slide saying let's say if there's a tumor in the parathyroid and there's too much parathyroid hormone being released, this would be hyperparathyroidism. So you have the osteoclast activity being increased. So that means that there would be too much bone resorption, and that means that the bone density will decrease. And this condition is known as osteostasis fibrosis cystica. So the bones become demineralized. There's too much loss of calcium because it's too much parathyroid being released. Okay, we'll stop here, and I will see you on Thursday. I hope you enjoy now that we are deep into physiology, starting with the skeletal system. Any questions? Yes, please. Ashley, you have a question.
Can you hear me?
Yes.
Okay. So you said that parathyroidism is stimulated when there's low calcium, right?
You mean when the release of are you referring to the release of parathyroid hormone or PTH occurs when there's low blood calcium levels?
Yes.
Okay.
Oh, is that I'm kind of confused because you said for the other slide, can you go back to the other slide for a second?
Here?
No, towards the end.
Here.
Yeah, you said that it's hyperparathyroidism.
Hyper. So that means there's too much parathyroid hormone being produced, right? Because there's a tumor, let's say. So remember that the parathyroid hormone activates osteoclasts, right? Because you want calcium to be removed from the bone to go into the blood. So that means if there's too much parathyroid hormone being released, that means that the osteoclasts are super active, they're unregulated, they keep removing calcium from the bone, and therefore this leads to the demineralization. So a loss of calcium from the bone. So this can lead, the bones are weakened, they can be deformed, and this condition is known as osteostasis fibrosis cystica, and it's caused by too much parathyroid hormone being released and too much osteoclast activity being activated by the parathyroid hormone. Is that clear?
So does that increase the amount of calcium in the blood or something? Like, where does the calcium go?
Can you say that again, please?
Where does the calcium go since it's being like destroyed?
Goes to the blood.
It goes to the blood. Okay.
Yes.
Okay, thank you.
You're welcome.
Have a good
So,
day.
can you explain the role of the gut? Okay, so the gut, there's calcium, right? In the lumen. So we are eating, there's the food has calcium. So inside the gut lumen, there's a lot of calcium, hopefully. So what happens to PTH and also vitamin D? What they do is they promote absorption of calcium from the gut into the blood. So there's more calcium. What does it mean, absorption? That means that there's more calcium being absorbed from the lumen of the gut into the blood. Is that clear? So that's why the gut is so important. Okay, so the slide with the, this one. So you have access to these. So you want me to repeat the effect. Why is it the gut important? So the gut is important because the gut has calcium levels, right? Inside our gut. Because we eat, we ingest food with calcium. So we have calcium, a calcium source inside the gut. So the parathyroid hormone, what it does, it increases the movement of calcium from the gut into the blood, and that's known as absorption. So you get more calcium being absorbed from the lumen of the intestine going into the blood. Is that clear? So remember that this condition happens when there's the level of calcium is low. So there's three targets of the PTH: the kidney to prevent calcium from being excreted, the bone to wait a minute. This is the wrong slide. This is the good slide where the PTH, here's calcitonin. Here's the PTH, sorry. So if the blood calcium level is low and you want to increase, the PTH acts at the kidney to prevent the kidney from excreting calcium. It acts on the bone to increase osteoclast activity so that there's more calcium being released from the blood, from the bone and going into the blood. And then PTH also and vitamin D increases absorption of calcium from the intestine into the blood. So PTH and calcitriol help the calcium found in the gut lumen be reabsorbed into the blood. Yes, but I would take out it's absorbed, because whatever is in the gut is there because we ingested it. It comes with the food, right? So it's absorbed, not reabsorbed. It's absorbed from the gut into the blood. So in parathyroid, there is an absorption of calcium from the lumen in the gut to the blood to increase the level of calcium in the blood. Yes. Okay. Is that good enough? No more questions?
Professor, I have one more question.
Yes.
It said on slide 34 that prior to puberty, the bone growth is regulated by insulin that comes from the pancreas. How would that be different for people with type 1 diabetes that can't make their own insulin?
Wait, can you say that slower, please?
Sure, just give me a moment.
Amila, can you
So I was wondering if they said that prior to puberty, insulin helps with bone growth. But
Yes.
how is it different for type 1 diabetics that can't make their own insulin?
Oh, that's a very good question. So, well, not all diabetic patients or not all diabetic patients are diabetic from birth, right? So under these conditions, you would have to provide insulin. But insulin is not the only and not the major factor that induces growth prior to puberty. You have this growth hormone that is produced by the pituitary gland. This is very, very important for growth. You have insulin growth factor that's produced by the bone and the liver, and the thyroid hormone and parathyroid hormone. And insulin plays a role, but it's minor. These other hormones play a heavier role. But that's a very good question.
So you're saying that type 1 diabetics, like, so insulin therapy replaces the pancreas, and you're saying that other factors are more important than insulin alone.
Yes.
Yeah, and a follow-up question. It said insulin growth factor, bone and liver. It doesn't mean that the IGF comes from the bone and liver? I'm a little bit
Yes.
confused.
It's
Okay,
produced by the bone and by the liver.
I see.
IGF is produced by the bone and the liver.
Thank you so much.
Okay. Okay. That's it. No more questions? Okay, so see you Thursday. Bye-bye.